Nucleic acids are polymers used to store information inside the cell. The most well-known nucleic acid is DNA, or deoxyribose nucleic acid, shown here in its traditional double helix structure. Many non-biologists don't know that there is a second nucleic acid called RNA, or ribonucleic acid. Both DNA and RNA are polymers made out of similar but slightly different subunits called nucleotides. RNA is an essential partner with DNA in the critical process of protein synthesis referred to as the Central Dogma. This video tutorial will introduce the structures of RNA and DNA and the nucleotide subunits that form them. It will present how each is used in the Central Dogma, and finally we will connect the nucleotides that make up nucleic acids as an important energy currency in the cell. Nucleic acids, like DNA and RNA, are built from smaller subunits called nucleotides. A basic understanding of nucleotide structure and how they bond to form RNA and DNA will be useful in your continued study of biology. There are two types of nucleic acids. DNA is made from the pentose sugar deoxyribose, and RNA is made from the pentose sugar ribose. Notice that each molecule in a strand is made from similar subunits. These individual subunits are called nucleotides. Let's take a closer look at a single nucleotide to learn more about its structure and diversity. This single nucleotide can be seen to be made of three different parts. On the right, the nitrogenous bases determine the sequence of the resulting nucleic acid. There are 5 different bases, divided into purines with 2 rings and the pyrimidines with 1 ring. The purines include adenine and guanine. The pyrimidines include cytosine, uracil, and thymine. The first letter in each pyrimidine spells the word cut, which can be used in the phrase cut the pyramid in half to remember which bases are pyrimidines. The second part of a nucleotide is the pentose sugar. The pentose sugar comes in 2 flavors and determines if the nucleotide will be used to make RNA or DNA. Ribose sugars have a hydroxyl group on the number 2 carbon, called the 2' carbon. The 3' carbon, located here, and the 5' carbon, located here, are also important in the pentose structure. Ribose nucleotides are only found in RNA. Deoxyribose is almost the same structure as ribose, but as the name implies, it is missing an oxygen. This may seem small, but it's very important to the proteins that make and use nucleic acids, and they can easily discriminate between ribose and deoxyribose based upon the presence or absence of that oxygen. The third part of the nucleotide are the phosphate ions. Since phosphate has a negative charge, phosphate groups regulate the charge of the nucleotide. They also regulate its energy level. We'll explore that feature of nucleotides later. Nucleotides can have a mono, di, or triphosphate groups attached to their pentose sugars. This image contains several new structures of different nucleotides made from different parts. See if you can name them or at least write their abbreviation. Pause the recording until you are ready to proceed. Let's see how you did. This first nucleotide has a ribose sugar, 3 phosphates, and an adenine base. So we'll label it ATP for adenosine triphosphate. This next example has a deoxyribose sugar and a cytosine base, as we can tell from our hints here on the corner. So we'll label this one C. We'll put a lowercase d in front of it. The lowercase d is used to indicate that this is a deoxyribose sugar. The ribose nucleotides don't have any prefix in front of them. So we have DC for cytosine, and this one only has 1 phosphate, so we'll call it monophosphate. This example has a ribose sugar, so there's no prefix needed. It has 2 phosphates, and it has a uracil base. So this will be UDP for uridine diphosphate. Our fourth example, again, has a deoxyribose pentose sugar. It has a guanine base and 3 phosphates. So we'll label it as DGTP, or deoxyguanosine triphosphate. Another question that you might want to ask yourself is, would this nucleotide be used to make a molecule of RNA, DNA, or could it be used in both? Although the nitrogenous base, guanine, is used in both DNA and RNA, this nucleotide is missing the hydroxyl group at the 2' carbon. That means it's a deoxyribonucleotide, and so it would only be used to make DNA. In our fifth structure right here, we ask the question, is this a pyrimidine, a purine, or neither? The correct answer is that it is neither. Although it has 1 ring structure, this is a pentose sugar, and the pyrimidines and purines refer only to the nitrogenous bases. Hopefully, going over these exercises helped you apply some of the principles you learned in an earlier slide, and help you see how the information might be used in the future. Covalent bonds form in a condensation reaction between nucleotides when a 3' hydroxyl group from a sugar reacts with a 5' phosphate group from another nucleotide. The new covalent bond is called a phosphodiester bond. Water and the phosphate ion are released. Phosphodiester bonds create a strand with a repeating sugar-phosphate pattern called a sugar-phosphate backbone. The bases project out from this backbone like teeth on a comb. This leaves them open to associate with other strands of nucleic acids. Let's use what you've learned in previous slides to identify this particular polymer. Is this a DNA or an RNA strand? Looking at the structure, again, you can see that there is a hydroxyl group on the 2' carbon. That would make this an RNA strand. Now let's review what we have learned about nucleic acid polymers and their nucleotide monomers. There are 2 types of nucleic acids, deoxyribose nucleic acid and ribose nucleic acid. Nucleic acids are polymers composed of nucleotide monomers. Nucleotides differ from each other in 3 parts. The first part is nitrogenous bases. The nitrogenous bases are adenine and guanine, which are purines, and cytosine, thymine, or uracil, which are pyrimidines. They also differ from each other in the type of pentose sugar. They can either have a deoxyribose sugar or a ribose sugar. And nucleotides will differ in their phosphate number. They may have 1, 2, or 3 phosphates attached. RNA nucleotides have a 2' hydroxyl group, and DNA nucleotides do not. Nucleotides can form a long strand by a condensation reaction between a phosphate group and a hydroxyl group. The covalent bond is called a phosphodiester bond. Now let's move on to our next concept. We just learned how nucleic acid strands can be built by linking individual nucleotides together with covalent phosphodiester bonds. Now we will see that 2 strands can also associate together using hydrogen bonding between base pairs. This forms the classic double helix so often associated with DNA. Proteins inside the cell can bind to major and minor grooves created by the double helix. The nitrogen bases of nucleic acids have polar functional groups that can participate in hydrogen bonding. Hydrogen bonding is a weak type of bond created by the attraction of partial opposite charges. These hydrogen bonds allow two strands of nucleic acids to come together to form a double helix. Since the interaction involves two nitrogenous bases, the resulting structure is called a base pair. This structure illustrates 4 base pairs. Hydrogen bonds between base pairs are illustrated as dotted lines to show that they are less permanent and more easily broken than covalent bonds that hold the sugar-phosphate backbone together. From this illustration, we will learn that base pairing is not random between nucleotides. It follows a set of rules called the base pair rules. For example, the functional groups of guanine are only able to hydrogen bond with complementary functional groups from the cytosine base. Guanine-cytosine base pairs form 3 hydrogen bonds between them. Adenine can form base pairs with thymine in DNA or uracil in RNA. Either base pair is only capable of 2 hydrogen bonds. If we look at the purple DNA strand on the left, notice that the top of the strand is structurally different from the bottom of the strand. To clearly identify one end of the strand from the other, scientists use the term 3' to describe this end and 5' to describe the other end. The terms are derived from the exposed 3' ribose carbon at one end and the exposed 5' ribose carbon at the other end. Often, but not always, the last phosphate group can be used to identify the 5' end. Now take a look at the pink RNA strand that is paired with our DNA strand. Can you identify the 3' and 5' ends? If you did it correctly, you may have noticed that the RNA strand runs in the opposite direction of the DNA strand. When nucleic acids form a double helix, they form an anti-parallel orientation. This means that the two strands run alongside each other, but in opposite directions. Here, we take a closer look at the molecular structure of the base pairs to understand why only certain pair combinations are possible. Remember that to form hydrogen bonds, there must be a partial positive charge and a partial negative charge. For each hydrogen bond shown in this image, you may want to pause the video and see if you can correctly identify the location of partial positive and negative charges in each bond. Here is the correct identification. With hydrogen bonds circled in green and partial negative atoms like oxygen or nitrogen circled in yellow. Notice that in each correct bonding pair, there is a green circle opposite a yellow circle. Now, look at the circles when incorrect mismatches occur. Now, look at the circles when incorrect mismatches occur. The yellow, yellow, and green, green pairs will actually repel each other and keep thymines from pairing with guanines and cytosines from pairing with adenines. Here is another example of mismatches that we will call ring mismatches. Note that in the correct pairing, whether it is an AT pair or a GC pair, the distance between the backbones is always 3 rings. This keeps the double helix the same diameter. Purine-purine and pyrimidine-pyrimidine mismatches, like the one shown here, not only mismatch partial charges, but their spacing is wrong as well. Pyrimidine-pyrimidine mismatches, like cytosine-thymine, are too far apart because they only total 2 rings. Purine-purine mismatches, like AG, don't work because they have 4 rings between them, and so they're too close together. A previous slide illustrated a double helix with one DNA strand and one RNA strand. Such complexes do exist briefly during processes such as replication and transcription inside the cell. But typically, DNA strands base pair with other DNA strands. These form very long, stable complexes that stretch for millions, if not billions, of base pairs. RNA molecules are typically much shorter. They're also typically single-stranded. Still, single-stranded RNA molecules can double back on themselves and form short distances of base pairs. These structures are often called hairpin loops because they resemble a bobby pin. From this we learned that nucleic acids can exist in DNA-DNA combinations, DNA-RNA combinations, and for short stretches as RNA-RNA combinations. So far, we have been representing a double helix as a flat structure because it was simpler to illustrate certain concepts with a flat model. In reality, the double helix structures twist around themselves like this. Notice as the helix winds around, the nitrogenous bases stack on top of each other in a flat orientation, like steps on a circular staircase. In this model, the major and minor grooves of DNA become apparent. So this would be the major groove right here, and then this would be the minor groove. Major, minor. These grooves allow proteins to read and bind specific base pair sequences in the helix in order to carry out cellular function. So you can see here that there's a certain part of the base pair exposed in these grooves, and that's what proteins bind to, like this one. These images illustrate ribbon models of two different proteins and how they contact the DNA in the major groove. Now that we've learned more about the double helix and nucleic acid structure, let's review what we have learned. First, nucleotides from one strand can also hydrogen bond with a nucleotide from another strand. This results in base pairs. Base pairing is determined by a set of rules. Adenine always pairs with thymine, or in RNA it pairs with uracil, and together they form two hydrogen bonds between the bases. Cytosine always pairs with guanine, and they form three hydrogen pairs between the bases. A DNA strand has a front and a back labeled 3' and 5' according to the numbering of the pentose sugar. A double helix can be made from two strands of DNA, which is the most common form. We also see complexes of a DNA strand with an RNA strand, or short stretches of an RNA strand with another RNA strand. A double helix has two strands that twist together. The base pairs lie flat in the middle of the helix, and the sugar-phosphate backbones twist around each other on the outside. These strands run in anti-parallel directions. The double helix forms major and minor grooves in its structure. This is where proteins can bind and read the sequence to carry out cell function. Now that we've learned about the structure of both nucleotides and nucleic acids, we'll move on to discuss how nucleic acids function inside the cell as carriers of information. The central dogma is probably better described as the central theory of molecular biology. It includes three processes that are fundamental to cellular activity and the very nature of life. These processes are replication, transcription, and translation. This video tutorial will provide an overview of the central theory itself and then introduce each process individually. The primary function of DNA is to store information with its sequence of nucleotide bases. The encoded information contains instructions for when, where, and which proteins get created inside the cell. This unique collection of protein machinery can then carry out the intended cell functions needed for life. Because of its important role, the DNA in many cells is housed in a protective compartment inside the cell called a nucleus. When a cell divides, it is important that each resulting daughter cell receive a complete and faithful copy of all the instructions encoded in the DNA. Replication is the process by which a DNA double helix is copied to create two new DNA molecules. It is important to note that replication involves copying the entire DNA content in the nucleus. While the DNA is housed in the nucleus, the cellular machinery needed to create proteins is located outside of the nucleus in the cellular cytosol. The DNA itself is too large and sensitive to exit the nucleus. So, to allow protein synthesis to occur, the cell creates short temporary RNA copies of the DNA instructions. The process of creating short RNA copies from a DNA template is called transcription, and it also occurs inside the nucleus. Once transcribed, the RNA molecule undergoes some processing steps in the nucleus before being shipped out through large pores into the cytosol. These RNAs are similar to note cards that a student might make from a large textbook held in a reserve library. The note cards don't contain all the information in the text. It just contains what is needed at the time to produce a good report. These temporary notes can be lost, ruined, or discarded without affecting the original information in the text itself. Translation, our third process, uses the RNA instructions from the nucleus to assemble a protein from amino acid subunits. In summary, replication creates two complete copies of DNA from a single DNA template. Transcription is used to create short single-stranded RNA copies from the DNA template, and translation is used to create molecules of proteins from RNA templates. Now we'll take a closer look at each one of these processes individually, starting with replication. One of the first steps in replication is to split the two strands of the DNA molecule apart. The protein machine that makes a new strand is called DNA polymerase. The base pair rules allow each strand to serve as a template for building a new strand. In this model, the newly created strands have a light purple sugar phosphate backbone, just so you can distinguish them from the older DNA strands that served as the template. This model only shows 26 base pairs being copied, but it is important to know that replication occurs along the entire DNA molecule and can extend for millions of base pairs in either direction. As a process, replication only occurs just prior to the cell dividing itself into two daughter cells, so it's not very often. It's kind of a big deal, similar in significance to packing up your entire house and moving it to a new location. Transcription is the process of creating a short single-stranded copy of a small region of the DNA. It also starts with splitting the DNA, but in transcription, only one of the DNA strands is copied. The protein that creates the RNA copy is called RNA polymerase because it polymerizes individual ribonucleotides into an RNA molecule. The resulting RNA sequence, distinguished here by the pink-colored sugar phosphate backbone, is a complement to the DNA template from which it is made. This means that an adenine base in the DNA template will create a uracil base in the RNA copy. Uracil is the complement or opposite base of adenine. Similarly, a guanine base pair in the DNA would show up as a cytosine base pair in the RNA copy. It is important to note that unlike replication, transcription only copies a small region of the DNA at any one time. Transcription is a process that happens all the time in cellular life, so it's similar in frequency to all the household chores you might do in your apartment, like cooking dinner, washing dishes, or doing laundry. Once an RNA molecule has been transcribed, it undergoes several processing steps before it is sent into the cytosol. Short regions inside the RNA molecule called introns are pulled out and removed. A polyadenine tail is added to the 3' end, and a guanidine cap is added to the 5' end. Details on these processes and others will be explored more fully in future lessons. For right now, it is enough to know that these processing steps must occur before the RNA is able to enter the cytosol. We will now move into our final process of translation. Translation is a more complex process than either transcription or replication because it involves translating information from the nucleic acid language, written in nucleotide letters, to the protein language, written in amino acid letters. The translated sequence of amino acid letters is determined by a messenger RNA, or mRNA for short. Unfortunately, there are only 4 nucleotides in the mRNA sequence, but there are 20 amino acids in the protein language, meaning that if each nucleotide designated only 1 amino acid, there would be 16 amino acids left out of the translation process. Fortunately, the messenger RNA is not read 1 base at a time, but in groups of 3 bases called codons. Each codon is matched, or rather complemented, by an anticodon on another RNA molecule called a transfer RNA, or tRNA for short. Individual tRNA molecules line up their amino acids according to the sequence in the messenger RNA. Each transfer RNA is loaded with specific amino acids that match its anticodon. The ribosome is a large cellular machine made up of both proteins and RNA. This third type of RNA is ribosomal RNA, or rRNA for short. The ribosome breaks the bond holding the amino acid to its tRNA sequence and reattaches it to the adjacent amino acid to form a chain of amino acids. This resulting unfolded chain is called a polypeptide. It is called a protein only after it folds into its functional protein structure. At the end of it all, translation can be summarized as the process of creating a protein from RNA templates. So in the process of translation, we see that not 1, but 3 different types of RNA are needed to complete the process. Messenger RNA forms a template strand for lining up the tRNA molecules. tRNA molecules match up the codons in the mRNA with specific amino acids, and ribosomal RNA forms an important part of the ribosome structure. In our model, we used a simple shape to represent tRNA. The actual structure looks more like this. Note that each type of RNA is still single-stranded and still made from the same 4 ribonucleotides. They only differ in their sequence and in the role that they play in the translation process. Now let's review what we have learned about the Central Dogma processes. We first learned that replication is where a DNA double helix splits and each strand serves as a template to create a new helix. Thus, 1 molecule of DNA becomes 2 molecules of DNA. Replication occurs in the nucleus, and it uses a protein called DNA polymerase to carry it out. Our second process was transcription. Transcription occurs when the DNA helix splits, and 1 strand serves as a template to make a complementary RNA molecule. Thus, DNA is used as a template to make a single-stranded RNA. Transcription also occurs in the nucleus. It uses RNA polymerase. Although the name RNA polymerase is similar to DNA polymerase, these are 2 different protein machines. There are different types of RNA made by transcription. mRNA, tRNA, and rRNA are all different types of RNA determined by their function. All of them work in the process of translation. Translation uses a ribosome, mRNA, and tRNA to build a polypeptide sequence, which then folds into a functional protein. Translation occurs in the cytosol. You should now have a better understanding of how nucleic acids function inside the cell. We have covered the structure and function of both DNA and RNA previously. Now, let's compare some of the similarities and differences between these two important biological molecules. Both DNA and RNA have a pentose sugar. However, DNA is made exclusively of deoxyribose pentose sugars, while RNA contains exclusively ribose pentose sugars. Both DNA and RNA have nitrogenous bases. In fact, the adenine, guanosine, and cytosine nitrogenous bases are identical for both molecules. DNA carries the nitrogenous base thymine. In RNA, however, uracil replaces thymine. DNA is typically double-stranded. RNA, on the other hand, is typically single-stranded, although that strand can bend back on itself and form short hairpin helices. DNA is typically much longer than RNA molecules. DNA molecules can stretch for millions to billions of base pairs. RNA, on the other hand, is generally much shorter, stretching for dozens to thousands of base pairs. Both DNA and RNA are used in the storage of information inside the cell. DNA forms a permanent information storage site and is housed within the nucleus. RNA is made in the nucleus, but its primary function takes place out in the cytosol, making proteins. It's more of a temporary information storage. RNA molecules are made, transferred to the cytosol, and then often degraded. DNA is made through a copying process called replication, in which a DNA template is split into two strands, and both strands are copied. RNA is made through the copying process of transcription. The DNA molecule is split and used as a template, but only one of the strands is copied to make the new RNA molecule. This concludes our comparison of DNA and RNA. In other lessons, we have learned how nucleotides can be added together in long polymer strands to form nucleic acids. Nucleotides also play an important role as an energy storage molecule inside the cell. We will start our exploration of nucleotides as energy currency by examining the structure of ATP. We'll then see how ATP can be used to perform work in the cell. This image shows the molecular structure of adenosine triphosphate, or ATP. The energy portion of ATP is contained within its triphosphate group. The ribose sugar and adenine base only serve as handles for the molecule to be recognized and held by necessary proteins. Each phosphate in the triphosphate group carries at least one negative charge, and at pH 7, the last phosphate, called the gamma phosphate, actually has a -2 charge. This creates a high concentration of negative charge in a relatively small space. And since these negative charges repel each other, it requires quite a bit of energy in the phosphate bonds to hold them together. In fact, most molecules could not maintain the -4 charge even for a short time. The triphosphate group is able to exist because it can spread the extra charge to each of its many electronegative oxygen atoms through a phenomenon known as resonance. Despite the resonant effect, the triphosphate group is still pretty unstable, and if given the opportunity, it will readily transfer its gamma phosphate to another molecule, allowing it to separate all those negative charges. When water is used to break and reform the gamma phosphate bond, it is called ATP hydrolysis. At lower levels of biology, it is often taught that breaking the phosphate bonds in ATP releases energy. That is actually incorrect. Breaking bonds always requires an input of energy. The energy release comes when the phosphate forms new bonds in a more stable position away from the other negative charges. The transfer of a gamma phosphate group from ATP to another molecule, like this protein, is called phosphorylation. Phosphorylation raises the potential energy of the new molecule, allowing it to do work. In a protein like the purple one shown here, adding a phosphate causes the protein to make a conformational shift, or a change in shape. The transfer of a gamma phosphate from ATP to another molecule, like this protein, is called phosphorylation. Phosphorylation raises the potential energy of the new molecule, allowing it to do work. In proteins, like this purple one shown here, adding a phosphate causes the protein to make a conformational shift, or a change in shape. These structural shifts can perform work, like the movement of this motor protein along the filament. Note the presence or absence of phosphate in each structure. Phosphorylation can also be used to move molecules across the cell membrane using phosphorylation-powered conformational shifts in transport proteins. The conformational shift in this protein changes its orientation in the membrane. These are just two examples of the many conformational shifts induced by phosphorylation in order to perform work in the cell. In small molecules, the phosphate group serves as a good leaving group that is easily replaced by other molecules. For example, the amino acid glutamate cannot react with ammonia to form glutamine because it does not have sufficient potential energy. However, it can receive a phosphate from ATP to create the high-energy intermediate glutamate phosphate. Ammonia is now easily able to replace the phosphate to create glutamine. Although I have limited our discussion so far to adenosine triphosphate, the other ribonucleotides, including guanosine triphosphate, thymidine triphosphate, and cytidine triphosphate, are also used in the cell for energy transfers, albeit somewhat less frequently. The deoxyribonucleotides, not shown here, tend to only be used in the synthesis of DNA. When ATP phosphorylates a protein or other molecule, it generates adenosine diphosphate. Eventually, the phosphate group is released from the protein as an inorganic phosphate, or PI. This makes room for another round of phosphorylation. The resulting phosphate ion and ADP are both transported into the mitochondria, where a special membrane protein called ATP synthase puts them back together. The newly formed ATP travels back out to the cytosol to do more work. Now, let's review what we have learned. The phosphate groups of ATP are easily transferred to other molecules to reduce charge localization. Energy is released when new bonds are formed in ATP, not when the ATP bond is broken. The phosphate structure is stabilized by resonance, otherwise it could not hold so much negative charge. Phosphorylation is the transfer of a phosphate to another molecule. When added to proteins, the phosphate usually causes a conformational shift. When added to molecules, the phosphate makes a good leaving group that allows new chemical bonds to form. ATP is a ribose nucleotide. GTP, CTP, TTP, and UTP are also used for energy, but less common. Deoxy-ATP, deoxy-GTP, and other deoxynucleotides are only used to make DNA. Finally, inorganic phosphate, PI, enters the mitochondria where it is added to ADP to regenerate the ATP lost as the cell does work. This video tutorial is part of the Biology Education Animation Research Project carried out at Brigham Young University, Idaho in the Biology Department. This video was produced and narrated by Dr. Stephen Christensen, Director of the program. Would you like to contribute? Contact Dr. Christensen by email at christensens@byuid.edu.